Welcome to the world of bioinformatics, where science and healthcare are used to transform lives. In this video, we will embark on the core concepts of bioinformatics and answer fundamental questions such as, what is bioinformatics, and when are these tests performed? Most importantly, we will shed light on how bioinformatics is pivotal within NHSBT. Bioinformatics is a relatively new field which became widespread after the Human Genome Project was completed. The vast amounts of data created needed to be sorted and decoded. Bioinformatics involves a combination of statistics, maths, computer science, and biology, and is used to develop tools to interpret complex biological data in many forms. Bioinformatics within NHSBT is used to develop, automate, and streamline analysis pipelines, and uses algorithms and statistics to analyze DNA sequencing data from patients, donors, and microbiological samples to improve patient outcomes. It is also used to answer specific questions such as, have efforts to increase minority ethnic donors been successful? Genomics, the study of the entire genetic makeup of an organism, uses bioinformatics to uncover the unique genetic signature that makes you you. This could include functional, structural, or comparative analysis. NHS Blood and Transplant use genomics to match patients to potential blood and organ donors by analysing genetic compatibility, while bioinformatics streamlines the process with algorithms, ensuring efficient transplants. NHSBT uses genomics to create human leukocyte antigen and human erythrocyte antigen profiles, which can gauge the degree of compatibility between individuals. Donor antigens must be similar to patient antigens, otherwise, patient antibodies will recognise these donor antigens as foreign, which will result in the cell being destroyed by white blood cells. Therefore, greater compatibility will lower the risk of rejection and ultimately improve the patient outcome. Now let's have a look at an example when genetic testing is used. NHSBT use a wide range of serology tests which make up the majority of investigations. But for rare blood groups, reagents may not be available, or if a patient has a complex mixture of antibodies, it can be difficult to identify the specificity. DNA sequencing is performed when serology techniques have been exhausted, or when a rare or novel phenotype is suspected. Genetic testing is also used in organ transplantation when matching donors with patients to reduce the risk of graft rejection. DNA testing produces vast amounts of genetic data that is processed and interpreted by expert bioinformaticians and genetic scientists. Newly produced sequence data is compared to the data already accumulated in databases and bioinformatic libraries, using bioinformatic tools, specialist software, and a variety of programming languages. These tools can be combined to create a bioinformatic pipeline. Patient DNA sequences are aligned to a reference genome which allows variants to be detected. The genetic basis of known blood groups is used to predict a majority of clinically significant red cell phenotypes. Variants identified that are not part of a known blood group undergo further investigation by our senior research scientists. A real example of a patient experience includes the case of a severely ill 83-year-old female living in Croatia. She had been admitted to hospital requiring an urgent blood transfusion. The laboratory were unable to resolve the patient's complex mixture of antibodies, therefore samples were sent to NHSBT International Blood Group Reference Laboratory for genetic testing, which revealed the patient to have a very rare blood type known as YTA negative. Incompatible blood was transfused to the patient due to low risk of reaction with the YTA antigen. However, they experienced a transfusion reaction. The discovery of the patient's blood group allows informed patient management for future transfusions and the provision of YTA negative blood.
Bioinformatics and genomics is going to have a very profound effect on NHS blood and transplant. Specific examples include array genotyping. So NHSBT has actually set up a world first genotyping service, and they've already typed 90,000 donors through that for all of their red cell, platelet and leukocyte antigen types. And this has been done in collaboration with the NIHR Bioresource Strides project. They're also in partnership with NHS England now typing 18,000 hemoglobinopathy, sickle cell and thalassemia patients from England. And so how is this going to affect their service delivery? Well, first of all, it's going to dramatically simplify the challenges of blood provision. One example is that a few years ago, a young man named Oscar needed a transplant, and I think we had many thousands of people volunteering to be screened because he had a particularly rare HLA type. It was 1 in 8,000 people. So in Strides, which is 90,000 donors, we've actually found 8 active donors with that HLA type through use of genomic data. Furthermore, once we type all of these donors and patients, it means that they'll have very dense antigen types available, which means that NHSBT can use bioinformatics and machine learning algorithms to implement a policy of precision matched blood, which minimizes the risk of immunization to the patient and also optimizes the use of blood within the organization. So these are two very big fundamental changes across the service in how blood products are used, and they'll be based on genomic data and machine learning and bioinformatics.
For more information about the services offered by NHSBT or the wider genomic strategy, please visit our website.